The overall goal of the following experiment is to study functional regeneration of intraspinal connections in organotypic spinal cord co-cultures using multi-electrode arrays, so-called MEAs. This is achieved by culturing two transversal spinal cord slices next to each other on MEAs to mimic intraspinal connections between different spinal cord segments in vitro. The slices grow, and within a few days in vitro, fuse along the sides facing each other. As a second step, lesions are performed after different timeframes in culture, which leads to a complete separation of the slices. Next, the cultures are left in the incubator for at least 2 weeks in order to give the spinal networks time to regenerate. The activity bursts between the two slices show that cultures lesioned at an early age show a high potential for functional regeneration, whereas this ability is clearly reduced in cultures lesioned at later ages.
The main advantage of this technique over existing methods, like slices cultured on membranes, dissociated cultures, or in vivo assays, is that the combination of organotypic cultures with multi-electrode arrays allows us to assess functional aspects of regeneration in a microenvironment of the spinal cord with direct experimental access.
To begin, fabricate or purchase pre-made multi-electrode arrays like the ones shown here. Sterilize the multi-electrode arrays by rinsing them for 30 seconds twice in 100% ethanol, once in 70% ethanol, and then twice in distilled water. Once dry, put 10 to 12 arrays in a glass petri dish and close the lid. After you have autoclaved the arrays for 20 minutes at 120 degrees Celsius, place each multi-electrode array into a separate sterile 35mm petri dish. Take a cooled pipette out of the freezer.